guys. Hello.
Hi,
Hi, how are you?
good. How are you? How is the
Good.
sound?
What's there?
How is the sound? Is it okay? Can you
Yeah,
hear
it's
me well?
fine. Yeah, I can hear you well.
Oh,
Right. Give a few more minutes for other people to join.
Hi, Nana.
Hey, Maureen, how are you?
I'm fine, thanks.
Do you hear me?
Hello.
Hello. Do you
hello.
hear me?
Uh,
Do you
yeah,
hear
but
me?
but I put louder.
Oh, okay. I guess it's the music. The music might be blocking it.
Yeah, it's a bit
Yeah.
too soft. Yeah.
Oh, okay. All right.
But the mic is not loud.
We'll wait another 5 minutes and then um,
Yeah.
just a minute. Hey, guys. Welcome everybody for now. We'll wait another 5 minutes and then we'll get started, okay?
Okay.
Hey, Jean, welcome.
Hey Kentucky homeowners, if you want to run your AC also.
Hey, Jean, welcome.
Thank you. Good morning.
Good morning. How are you?
At least. How are you today?
I'm well, thank you.
Good.
All right. 3 more minutes and then we'll get started.
can I get a collection of your collection of songs?
Oh yeah, it's Very Mamo.
What's the name?
Very Mamo.
Okay, I'll find it.
Yeah, I'll send you the. All right, guys, welcome to everybody to this conversation. The Hearts and Kotoko match is in 2 hours, so we kind of have to get going because I think at 3 o'clock everybody's just going to bail. Welcome so much, and I'm glad we could put this together. I have 4 Ghanaian scientists here, young Ghanaian scientists, and we are going to talk biosciences today and how it can relate to the development of Ghana and maybe even to a greater portion to the whole African continent. Under the shadow of COVID, I think this is an important discussion to have, and I hope this won't be the last one. I won't talk anymore. I'll just open up the floor to our participants, and I'll start with Jean. If you want to introduce yourself and what you're all about.
Sounds good. Good morning, good afternoon, good evening, everyone. Thank you for joining us. My name is Jean Bronte. I'm just finished up my PhD with the University of Kentucky in the United States. I'm a virologist. Well, I'm trained as a virologist in a biochemistry department. What I'm about, I got a fascination with viruses very early. I actually grew up a germaphobe. I was a little afraid of things I couldn't see. And I remember when I was younger, my siblings would tell me to wash my hands. So I took that very seriously because I'd seen all those commercials about how you could get cholera and get, diseases from pathogens that are beyond us being able to see. So I got a fascination from viruses at that age. I also really enjoyed science, and I was that kid who asked all the questions. And so as I was choosing my career, it almost was intuitive that I would end up in something related to microbiology. So I leaned into my fear. I'm not scared of viruses as much anymore. At least that fear that made me stay away now became a fascination that made me drawn to microbes. But yeah, that's kind of my background. I did my undergrad here in the States, and I also just finished my PhD here in the States. And anything else, please go ahead and ask me in the chat or anything else. But that's a little bit about me.
All right. Well, Jean, thank you so much. If you could make your light a little stronger, if possible, so people can see your face.
Yeah,
If not, but we can hear you great.
I have my light on. It's just a really gloomy day today.
Gloomy day. Yeah. No problem. No problem.
Okay. Sorry?
Yeah. Oh, no problem. I'll give the floor now to Regina. And am I saying it right? Some people say Regina, some people say Regina. And how do you pronounce your first name?
And that's the confusion for me. I mean, in Ghana, my family, my friends, everyone calls me Regina. And then I moved to the UK first, and everyone was calling me Regina. Even here in Hong Kong, so they call me, I think I've gotten used to Regina now, so yes.
Okay, yeah.
Hello, everyone.
Yeah.
Should I go ahead?
Go ahead, yeah, please.
Hi, good evening from Hong Kong. My name is Regina Kwarten. My friends call me Reggie. I'm a PhD student at the Hong Kong Polytechnic University. I'm Ghanaian, in case you didn't know. So I'll talk a bit about my research here at the Hong Kong Polytechnic University. So I'm what you call a myopia researcher. Basically, I study the genetics of myopia, right? So I'll talk a bit about my research, which is myopia. What is myopia? I don't think it's a strange topic, so I'm sure many know about myopia. Myopia is a very common eye disorder, which occurs when the eyeball is elongated. So people with myopia, they can see near objects clearly, but they can't see distant objects. The problem with myopia is that it's extremely prevalent, especially in this part of the world, in countries in East Asia, like China, South Korea, Hong Kong, Japan. The prevalence rates can reach as high as 90%, and it affects a lot of the young population. So over here in Asia, they describe it as a myopia boom among the young population. So myopia is what we describe as a complex trait, meaning that it's affected by both environmental and genetic risk factors. I'm particularly interested in the genetics of myopia. So on the genetics, what we've been doing is using a technique known as genome-wide association studies, or GWAS, to study genetic predisposition to myopia. So I'll explain a bit about myopia. There are so many things I need to explain.
I'll cut you short right there. We'll get to the second round, and I'll give you more time to talk about that. I just wanted to do a quick introduction.
All right, so that's a
all right. So yeah, so just a quick intro of who you are and what you're about. Yeah, and then we'll come back to that. Okay.
All right. All right.
Yeah. So Aloysius, you're next. Can you tell us just a short intro about who you are and what you're about?
Yeah. Hello, everybody. Can you hear me?
Yeah, we can.
Okay. I'm Aloysius Jibodiloglo. I'm a researcher, and I've been in the field of working on Borreliosa for some years now. And over the years, I've developed more and more interest into neglected tropical diseases. And I've been doing this, and based on that, I've expanded into immunology, which is my main interest, to understand how people infected with these diseases can be, managed to get better clinical outcomes. I'm a PhD student registered at University of Surrey. I worked there for a while as a research assistant, and then I came back to Ghana to complete my PhD. So my PhD briefly is looking at the pathogenesis of Borreliosa from the perspective of diet. So I'm using some tools in nutrition and some tools in immunology to answer the question, and also some tools in public health. And also, I'm married with 2 kids, and I'm a Catholic. Thank you.
Okay, thank you so much. So our last panelist will be Justice. And Justice, do you want to tell us a little bit about yourself?
So hi, my name is Justice Kofi Wachiapia. I currently live and work in London, UK, but I'm very Ghanaian, wholly Ghanaian. I trained primarily as a medical doctor, and then after medical school, after 2 years of practice, branched into science. I started off my career doing my housemanship, combined medicine and research in microbiology, infectious diseases, basically working on TB and Borreliosa disease. As Aloysius also works in Borreliosa, and then decided to take a PhD, or more like got an opportunity to take a PhD in infection and immunity, but I looked more into vaccine development. And so my work was basically, my PhD was mostly on Borreliosa disease, developing a vaccine against Borreliosa disease. But along the line, I got back into TB through some research grants that I was gracious enough to have, I was fortunate enough to have won. After my PhD, during my PhD, COVID broke out. And so I think some vaccinologists were needed to get involved in the clinical trials and so on. So I got co-opted onto the team working on the clinical vaccine and a number of other vaccines, conducting clinical trials for those. So I started working with the National Institute of Health Research in the UK at the University College London Hospital. I've been there since, but I still maintain my ties to my Borreliose and TB work at the St. George's University of London. So I sort of run both while still doing some minimal but clinical practice on the side as well. So that's basically what I do.
Well, thank you, and welcome all of you, and thanks for doing this. I really do appreciate that. We'll go back to Jean, and I'll ask you why the basic sciences. Why not medicine? Why not anything else? Why not computers? But why the basic sciences? And I know your work was based on respiratory viruses. So why that?
That's a great question. I'm very much Ghanaian. I want to point out, I grew up in Ghana and was in Ghana. Well, I shuttled between Ghana and East Africa until college, where I moved here. And I remember when it was time to apply for college, even though my interest was generally in what I now know are the basic sciences, I thought that I had to go to medical school. I thought that was really the only way I could do research. I'll share a secret with you, and I guess this is an open secret now because everybody's gonna know. I am very squeamish. I see blood and I run away. I haven't done any animal research. I do research in cell culture. And so anyone who's familiar with the research environment and basic sciences, that you're doing work using whole animals. So I saw that not only was I drawn to basic sciences, but there was also a gap. There's a little bit of a vacuum in terms of people who were willingly going into basic sciences as a research career. For some, I know, for example, at the University of Ghana, if you apply for medical school and then you don't get in, then you do biochemistry. That kind of is the narrative, or appeared to be the narrative, which unfortunately I don't think is accurate or should, it's happening, but I don't think should be the case, right? We shouldn't, getting into basic sciences requires high level performance, and it's not something that should be looked at as something that you get into if you didn't get into your number one choice of practicing clinical medicine. There's a little bit of a gap there. I just knew, even though I didn't have a lot of examples, or I personally wasn't aware of a lot of examples in research. You hear of the Einsteins, and maybe you would hear of Marie Curie, but I didn't really know that research was a career option for me. All I knew was I wanted to kind of learn. And so it was during undergrad that I figured out, wait, there's a where you can do research and you can teach, but you can also just focus on the basic science that develops a framework for medicines and for access to health. So that's what inspired my reroute, or my desire to get into the basic sciences. And the reason that I was even interested in basic sciences starts with our own environment. So the goal here in basic sciences a lot of the time is to identify some kind of target, right? So you look at a giant cellular process, and it might look like a lot of things that are worth getting into just to know. For example, if you look at a signaling pathway, you might think, why do we care about this particular kinase or this particular enzyme, and how it cascades into something going into the nucleus and doing some really minor thing. It looks like that from when you look at it without the context of a big picture. But what I really wanted to get into was go in and look at cells on a very microscopic or beyond microscopic level. Let's look at the signaling that's happening. Let's look at the biochemistry that's happening. And what that hopefully would allow us to do, and what my thought was that'll allow us to figure out what is a dispensable target. What is something that if I had this medication and I targeted it in a host cell, it would prevent infection, but without poisoning our cells. And I think that's something that's really important to know, because that is the framework and the basis of understanding how medications work and developing new medications, identifying new targets. So that is the inspiration behind why I got into basic sciences, and it's a booming field. We could always use lots of great minds in basic science, and so it's an exciting spot for me, and I'm enjoying it.
Thank you so much. You brought up a very interesting topic, and it's something I've often discussed with friends, and it's that you look at that whole environment in Ghana, and when people score really high on the university entrance exams, the first thought is that they will become doctors, and there's never a thought that these people should go into the basic sciences and become researchers. And unfortunately, that is sort of how it's been. But these people should be becoming the physicists and the physicists and biochemists like you, and vaccinologists and geneticists and molecular biologists, because it's on that basis that even medicine is born and progresses. Thank you so much for bringing it up. We will talk about that some more later. Regina, so let's go back to myopia. I mean, I wear glasses, so I know what you're talking about. So what drew you to that topic in particular?
Okay, so in my case, it's not necessarily the topic myopia, but the technique, as I was talking about, right? So I'm a molecular biologist, a geneticist. So basically using the techniques of molecular biology to understand diseases. So I was talking about genome-wide association studies, or GWAS, right? This is a technique in molecular biology where we use genetic variants to predict people who are at most risks of developing diseases, right? How do we do this? We basically study common genetic variants, or single nucleotide polymorphisms is pronounced as SNPs, right? So in a population, you will have people affected with a disease and those without it. So with this technique of GWAS, we can compare the frequency of these common genetic variants, right, in people affected with the disease or the cases, and those without it or controls. And we can identify SNPs, genetic variants that predispose these people to developing these diseases. So that is what I've been doing. I mean, I've been studying, and myopia is just one of the traits we can apply this technique to study. We can do it.
So what drives you is basically molecular biology techniques.
Exactly. I love genetics. Okay. So when I first met my supervisor, my supervisor is a geneticist by training, right? He studies not just myopia, he studies cancers and complex traits in general. So yes, what drives me is the genetics and then the molecular biology. And then we apply it to traits, to diseases. So we use these techniques to understand the genetics underpinnings of these diseases. So that's my drive, actually.
Yeah, so full disclosure, just before I moved to the US from Germany, where I did med school, I did a whole year in a genetics lab. Once upon a time, I almost became a geneticist. So I kind of understand where you're coming from with that.
That's great.
Thank you so much, and yeah. Go ahead.
I was going to continue, but
yeah, go ahead. Sorry. Sorry.
Okay. All right. So what I was going to highlight, the things we've done in my field using this technique of GWAS in myopia research, right? So using this technique, we've identified so many genetic regions and what we call genetic variants, which are associated with myopia. And with that, we identify more than 400 genetic loci, right? But the problem with my research, or one of the biggest limitation of my research, is that when we talk about association, it's not the same as causation, all right? So we need to move the field further. We need to we need to bridge this gap in the research, right? In order to identify actual causal genes, that is genes that are directly implicated in myopia development. So that is what I've been studying, actually. So in my research, I use the tools of molecular biology to understand the genetic causes of myopia. So in the lab, you find me doing a lot of molecular biology stuff, cloning, PCR, electrophoresis, Western blots. I'm using a whole lot of techniques. And in the moment, I'm trying to develop what we call a high throughput technology that we can use to screen all these, several hundreds of variants we've identified to be associated with myopia. So that is my research.
Well, thank you so much. And it's important to say that knowing these techniques really matter, because what you do underpins what Jean does. It underpins what Aloysius does. It underpins what Justice does.
Yes.
Through all this COVID thing, we all heard of PCRs and PCRs and PCRs, and that's basically the baby of molecular biology. In the next round, I'll come back to you and ask you to talk about a particular technique that has carried the world by storm lately.
All right. Okay.
Next, we'll go to Aloysius and ask you how you got to Buruli ulcer as your target. And also, I'll give, if you have any questions, just send them to me by the chat, and we'll get them to the panelists somewhere along the line. Hey, Aloysius, go ahead.
Okay.
So why Buruli ulcer?
Thank you. So I kind of stumbled into Buruli ulcer. That's how I started with it. My interest is mostly into how to solve a problem using simple techniques. So my first research experience was actually related to HIV co-infections. And when we're doing that study, I realized that some patients were defaulting to treatment, discussed it with the PI, and decided to do just a simple qualitative study to understand why they default. So that's been the mindset. So I worked in that for, let's say, 3 years, and then I jumped into Buruli ulcer.
I think we may be jumping the gun. Can you give people a little background about neglected tropical diseases and Buruli ulcer and that whole topic? Just a short discussion of it. Because I think that would.
Yeah. So neglected tropical diseases are a collective group of 20 diseases designated by WHO. And they are endemic in tropical and subtropical climates, and it's killed over 1 billion people. The problem is that about 70% of the cases are found in Africa, and we have scattered endemicity in our parts, the parts of West Africa and Ghana to be specific. And the another interesting thing is it affects mostly women and children who are less than 15 years. So it's basically diseases of kids. So Buruli ulcer is one of those diseases, and it has very serious effect on the skin. It can lead to disability, and it can also lead to lifelong disability. It also has mental health issues, which currently is being dealt with because those who are infected with the disease are mostly neglected and discriminated against. So the reason why I went into Buruli ulcer research was because one, apart from us, there's nobody really helping people infected with this disease. And the other thing is I can tell that I'm making an impact because we manage the patients, their lesion heals, they are happy, everybody's happy, they go back to their lives. And those who are from schools, students are able to continue with schooling. That is if they don't come with the worst stage of the disease. So the problem with NTDs is that it's underfunded. And because of that, it's difficult to get money to manage those diseases. And though control programs in our nations are doing the best they can, we need a lot of money to really combat it. For instance, HIV gets about 37% of the world's funding. And NTDs, that's a collective of over 20 diseases, gets about 0.6% of funding. So you can just tell the dichotomy between the two diseases.
And Aloysius, some of those diseases are not very expensive to treat, right? I mean, some cost like 20 cents per patient a year, something like that.
Yeah, scabies, for instance, you need ivermectin. You do mass drug administration and then it's cleared. So it's not really that difficult. I think the problem has to do with visibility and availability of information. That is why maybe we are not really getting the funding we need to make things happen.
So they are basically diseases of the unseen poor, you would say that.
Yeah, the unseen poor. Yeah,
the unseen poor. All right.
Yeah, because they live in poor, remote communities, which means that even if you are in Ghana, you are not likely to know those
to
places.
be seen. Yeah. I would have loved to show a picture of Bury also, but I think I won't because it's not very pleasant. But I would really recommend that everybody Googles it who doesn't know about that and look at the pictures. They are very disfiguring. An ulcer is basically a wound, and this is a disease where you get these wounds that are quite disfiguring. It spells B-U-R-U-L-I, an ulcer, and it's sort of incumbent on all of us to pitch in somehow to get this under control. And I kind of like the idea that I have two panelists who are involved in that. So Lucius will come and talk about that some more. I'll go to Justice now. Okay. And he is actually working on a vaccine for Buruli ulcer. And I must say that his work is very, very interesting, and the platform he's using, it's a very interesting platform, pretty much like what Novavax used to make their vaccine for COVID. It's just that his is actually a lot cheaper and not as protracted. And so, Justice, can you talk about that a little bit?
So yes, so the idea is to make cheap vaccines, which are accessible to everyone, not too expensive. Vaccines are currently quite expensive, and as you can tell, even during this COVID pandemic, one of the main issues has been the cost of vaccines and the complexity of vaccine development. If you're making a vaccine, you're interested in getting what we call antigens. So antigens are possibly parts of a virus or a bacteria which will be able to induce good immune response and give you protection. Traditionally, the way of making a vaccine, as we were taught in primary school or secondary school, is to just take the bacteria, the whole bacteria or the whole virus, weaken it or kill it, and then put it in the human being so that the person's immune system recognizes that virus or bacteria, builds all the soldiers or gathers soldiers to go fight it, knowing that this is a weak or a dead virus or a bacteria. And then in future, when the real or living bacteria comes in, the immune system is able to remember, recall, and say, so the word recall is very important in immunology or in vaccinology. Recall that, oh, we've seen this before, and this is how we tackled it the other time. It gathers the soldiers, and then it quickly goes to fight. Over the period, we have seen that there are complications in putting whole cell bacteria or viruses in human beings. You know, even if you've weakened it or you've killed it, it comes with some problems, especially in the era where we had diseases like HIV, diabetes, and so on, which reduced people's immune systems, the strength of people's immune systems. It meant that if you were putting in a weak virus or a weak bacteria, you really could not tell how weak it was, because it may be weak for a very ordinary person, for a normal person, but for someone with HIV, it may still be too strong, you know, and there were these considerations. So the way forward now has been to make vaccines using what we call subunit proteins. So you take a very small part of the bacteria or the virus, which you know the immune system is able to recognize. And that has been the challenge, identifying which part of a virus or a bacteria the immune system is able to recognize. And that's basically what I have been working on. So I've been trying to identify which parts of the bacteria that causes Buruli ulcer disease the immune system recognizes and is able to build an immune response to. And I've done the same for TB as well. Now, for Buruli ulcer, the interesting part is that it's not just the bacteria that causes the problem, but the bacteria also secretes a certain toxin, and that toxin is known to underlie all the effects, almost all the effects you see in Buruli ulcer disease. So if you're able to clear the bacteria, you have not solved the problem. You should also be able to clear the toxin to be able to solve the problem. And that is what has brought about the complexity of or the absence of a vaccine for it, even though there has been work ongoing for years and with scientists trying to develop a vaccine for it. We've been able to crack it. We have a candidate, which is sort of a combination candidate. It takes parts of the bacteria itself It also takes parts of the toxin, and then we identified what we call adjuvants. So adjuvants are basically chemical compounds or molecules which improve the immunogenicity. Immunogenicity basically means the ability of the immune system to recognize that protein or that antigen, or the molecule, the toxic molecule you're putting into the body. So we combine these, and we have a candidate which in mice gave almost 100% protection against Buruli ulcer disease. So that's very good prospects. Going forward, we managed to get some grants for it. We extended the technology to TB as well, and that's currently what I work on in St. George's. Aside my COVID work, we extended the technology into TB as well. Still working on that in mice and seeing how far that goes. And the most interesting part of it was that during the COVID pandemic, some pharmaceutical companies have latched onto this technology and this platform of using just subunit vaccines, subunit proteins, adding them to adjuvants or combining them with adjuvants to develop vaccines for COVID. And there are about 2 of such in the system. Sputnik is one of such. It has, well, not entirely, but it has something like that. The Novavax vaccine is purely that, basically. My method is cheap, but yeah. It brings a lot of prospects for this, and it makes me happy to know that what we do, the platforms, this is a cheap platform, makes very cheap vaccines, made very good vaccines, very, very safe vaccines with very, very, very good responses for the immune system and with good protection rates and efficacy rates.
But you don't use the ovary of the armyworm. I mean, that's really cool. You should do that.
Yeah. So they use the ovary of the army. But the intriguing part of it is that as part of the science, we're also interested in cost. So what I do is to use E. coli. E. coli, of course. E. coli is a bacteria. It's a bacteria that is common. It's everywhere. It's everywhere. Causes infections and so on in humans. But there are some E. coli which are not necessarily infectious in humans. So what I do is I take a bit of what Regina does. That's the genetics of it. I sort of change the genetic makeup of the bacteria, the E. coli, and then I get it to produce or to secrete the protein that I want. So basically, taking the protein genes out, or what we call the plasmid, out of the ulcerans bacteria, the bacteria that causes Buruli ulcer or TB, put it into the E. coli and cause the E. coli to secrete the proteins for me. There's an adjacent lab which is using plants, cigarette, tobacco plants. Which is using the tobacco plant to secrete these proteins in the same way. So I use E. coli, very cheap, very reproducible, very scalable to a very high extent, and then you get very clean proteins which you're able to develop your vaccine with. The pharmaceutical companies have the money and the platforms, and they want a quick way out, so they use quicker methods, which tend to be a bit expensive. But in future, I'm optimistic this method may catch up soon. And
it's very interesting. But Justice's platform, it's very similar to the platform Novavax is using, and which tells you that he could easily have made a COVID vaccine if
at the beginning, I think this was the simplest design. And I mean, the simplest design was just to put the virus, which is what the Chinese people did. But if we had the platforms,
their vaccine is a little questionable, but we won't get into that.
I don't want to get into the Cold War politics.
All right, but now we'll come back. We'll come back to you. We'll go back to Regina. And in terms of technique, there's one technique that is basically taking the world by storm since those 2 women won the Nobel Prize, and that's the technique of Cas9 CRISPR. And I know you admire those women greatly, and so exactly. So talk a little bit about the technique and talk about how it relates to certain diseases that afflict us, and especially sickle cell disease. How can we use this technique, CRISPR-Cas9, to maybe find a treatment for sickle cell disease?
Okay, all right. So this might take more than a minute for me to.
Go ahead.
Okay. So CRISPR is actually an acronym, and the full meaning is, I hope I get this right, clustered, regularly interspaced, short palindromic repeats. So that is CRISPR. Okay. So in simple terms, what is CRISPR? I mean, they are just DNA sequences that belong to viruses that infect a bacteria. Okay? So when a virus infects a bacteria, the bacteria tend to store or keep parts of the viral DNA so that upon subsequent attack, it quickly recognizes, the DNA sequences, and then it cleaves or cuts these sequences. So basically, that is what CRISPR is. They are just DNA sequences that belong to viruses that infect bacteria. Then there is case 9. The C in case 9 actually stands for CRISPR. So CRISPR associated protein 9, that is case 9, okay? So what is case 9? Case 9 is basically an enzyme, and its sole purpose is to look for DNA sequences which are complementary to the CRISPR sequences and then cleaves them or cuts them. So that's the sole purpose of case 9. So if you then join CRISPR to case 9, then we have the CRISPR case 9 technology. And potentially you are looking at a tool that can be used to edit the genome, right? So two researchers, the two women you talked about, Emmanuel
Charpentier and Doudna.
Yeah.
Yeah. Charpentier and Doudna.
Yeah. And then Jennifer Doudna, who is an American, the two met at a conference and they realized they're working basically in the same field. So what they did then was to collaborate. And then the work they did was quite revolutionary, and which won them the Nobel Prize. So what they did was they took the case 9 protein, which is a 4-complex protein, and then they re-engineered it into a 2-complex structure, which they called the single guide RNA. Now, what's interesting about this single guide RNA is that when it's joined together with the case 9, then it can cut DNA fragments at anywhere within the genome. So if you are interested in cutting or editing anywhere in the genome, all you have to do is to make sequences that are complementary to the single guide RNA. And with the case 9 attached to it, so what happens is that the single guide RNA just tells the case 9, right? You're going into the genome, and you, the case 9, you're going to make cuts, or you're going to edit this exact region in the genome and nowhere else, okay? So yes, so CRISPR-Cas9 technology is basically molecular scissors that are used to, make cuts and then edit the genome of the human genome, basically. So it's not, the real application of this technology is in what happens once we cut the genome or make these deletions. So 3 scenarios are likely to happen, right? The first scenario is when we make just a single cut in the genome. You make just the case 9 makes a single cut. And the thing is, the DNA will tend to repair itself, right? Using its own repair mechanism. So this method is called non-homologous enjoining. So the DNA will repair itself, but the problem is the repair process is very error prone. So while repairing itself, it introduces insertions, deletions, mutations. So at the end of the day, the repaired DNA is not exactly as the original DNA. So by that, you've been able to what we call disrupt or inactivated a gene, right? So that is case 1. The second case scenario is when we are interested in deleting a DNA fragment. So in this case, we can use 2 guide RNAs to delete, say, a large fragment of DNA, and then the guide RNA together with the case 9 will make these cleaves, and then we can basically cut these DNA fragments. So once again, the cell will repair itself once the the fragment is deleted using what I described as the non-homologous enjoining, right? So that's by in that situation we can knock down a gene or delete a gene region. The last case scenario, sorry, let me take a drink before I continue. It's getting so interesting.
I can imagine.
Yeah. The third scenario is when there is a faulty gene. There is a gene with mutations in it, and we want to sort of correct for that gene. So what we can do is we design a template, right? A template with the correct sequences. And then we can use the CRISPR-Cas9 technology to introduce this new template to correct or replace a faulty gene. So this is what we call the homology-directed repair. Are you with me up to this point?
Yes, I think so. We are.
Okay. Okay, so now we can look at the real-life application of this technology. So to do that, let's pick a disease like sickle cell disease, right? So sickle cell disease, I think many of our viewers know what sickle cell disease, a red blood cell disorder So I'm a biochemist. So first of all, let's look at the biochemistry of it, right? So the hemoglobin gene is defective in sickle cell, specifically the beta globulin gene, right? So what happens is the hemoglobin gene is made up of 147 amino acids. Each plays at specific positions, right? So on position 6, the amino acid there is glutamic acid. And the genetic sequence for glutamic acid is GAG, right? So GAG, these 3 letters, GAG, and then it will code for glutamic acid. So in people with sickle cell disease, there is what we call a single base substitution. So instead of GAG, the code now is GTG. So an A has been substituted for a T. Now, that has very devastating effects because that codes for a different amino acid, which is valine. Now, if you look at the structure of glutamic acid, it's quite different from that of valine. Glutamic acid is, it has an extra carboxyl group attached to it. Valine, on the other hand, has a hydrocarbon, an isopropyl group, right? So structurally, these 2 amino acids are quite different. And the kind of reactions they partake in are also quite different as well. So glutamic acid is hydrophilic, meaning it attracts water. And then valine is hydrophobic, so it repels water. So these contribute to the deformed shape or the defective shape of the red blood cells in sickle cell disease. So that's the biochemistry. Then we look at the genotypes, right? So sickle cell disease is what we call a recessive trait, meaning that you need 2 copies of the abnormal hemoglobin in order to develop the disease. So normally when you talk of sickle cell, the medical practitioner will tell you of the SS and then the AS, right? So the 2 SS represent the 2 copies of the abnormal hemoglobin. And then the AS, meaning you have a normal hemoglobin and 1 abnormal hemoglobin, right? So the genotypes present different in people. So those with AS, they tend to be what we call sickle cell traits, meaning they are just carriers. They don't suffer from any of the complications associated with sickle cell disease. Whereas those with the 2 copies, the SS, they suffer all the complications, the severe symptoms associated with sickle cell disease, right? So looking on the clinical, the therapeutic aspect of the CRISPR-Cas9 technology in sickle cell, if we are able to convert just one of the alleles, the abnormal alleles from SS to AS, then we can potentially convert these sickle cell disease patients to sickle cell traits or sickle cell carriers. Are you still with me?
Yes, yes.
Good. So to do that, we can use the CRISPR-Cas9 technology. And I'm sure many people are wondering, how do you do this? Okay. It's not magic. So what you can do is, we take the sickle cell patient, the SS, those with the SS genotype, we extract peripheral blood. And from the peripheral blood, we can harvest stem cells or progenitor cells,
yep.
right? Good. And then the stem cells, we can now use the CRISPR-Cas9 technology. Remember when I talked about the homology-directed repair, where you can design a template to replace or correct for a defective template, right? So we can use the homology-directed repair and use that to convert one of the abnormal S into an A, okay? So we can use that to edit the stem cells. And then the next step is we can then screen for the stem cells that were correctly edited, or the stem cells which have now been converted from SS to AS, okay? Now we harvest these stem cells, and then we can culture them and clone them, and later transplant them back into the sickle cell patient. So once it gets into the body, the cells mechanisms just take over, right? So once again, this is a very practical tool. And I think earlier this year in January, in the, University of Illinois in the US, they've used this technology to treat people with sickle cell disease and beta thalassemia. And just this morning, I was reading about a paper where they've used this technology to treat another rare genetic disease. I can't even pronounce the name, it's that rare. So yes, CRISPR has, when I get scared when I think of all the possibilities. with
You know what?
CRISPR. And
I think we should have another section where we just talk about CRISPR. What do you think?
that would be great.
I think we should. And
That would
just
be
to add
great.
to
That would be
that,
great.
just to add to that, before they they transfuse the stem cells, they usually will try and kill off the sickle cells that are already in the patient. So the patients would have a little chemotherapy before,
Oh
and then they
yes.
get
Oh yes.
transfused them.
Obviously,
And
it's transplantation.
there's also the possibility of using CRISPR to increase the amount of fetal hemoglobin, right? Because fetal hemoglobin allows the body to carry oxygen properly. So
Also,
that's another
this is just
possibility of that.
yes, one possibility
Yeah.
I talked about.
Yeah.
Yes.
Yeah.
So can I go on? Oh, I'm running out of time.
Yeah. Let's come back to you. And since we are running out of time. And,
Oh, okay. Then I'll
okay.
just pause here.
Yeah.
We will continue later.
Okay. So we'll go to Jean. Jean, so respiratory viruses. I mean, if anybody doubted how important they are, the last 15 months have told us that they are the class of viruses we need to be scared of the most. And I keep telling people are scared of Ebola, people are scared of HIV, but you need to come into contact with bodily fluids to get those diseases. Respiratory viruses, you can be in your somewhere and somebody coughs or breathes and you have it. And to me, they are the scariest of all the viruses that they are. And so can you talk a little bit about their importance, where research is headed in terms of treating them, and what you did basically in terms of your dissertation?
Perfect. No, you're definitely right. A lot of us hear about viruses and our thoughts go to Ebola and other emerging viruses like Hendra, Nipah, which broke out in India. So what makes respiratory viruses and even Ebola and those really deadly ones so deadly and diabolical is they prey on our social instincts. And so humans are social beings, right? We hold hands, we hug, we interact with each other. The one thing about Ebola or Nipah virus or anything like that is it's not airborne. It's not carried in droplets. If it's, if you cough and leave droplets on a surface, you may not be as exposed. But that's not the case for influenza. That's not the case for SARS-CoV-2, as we realize now. You as you pointed out, you can be sitting somewhere and then somebody coughs on a door handle or some droplets landed on a door handle and you think you're being careful, you're masked up, but then you touch a door handle and then you accidentally touch your face or some mucous membrane, and then next thing you know you have a deadly virus. So one thing that we definitely need to point out about respiratory viruses and what makes them so dangerous is definitely the airborne nature, right? The fact that you can inhale these viruses and they can lodge into anywhere in your airway and start infection and then prevent you from, wreck your insides in terms of your respiratory tract, making it very difficult for your body to absorb oxygen, which is what you need to function. So one thing that's interesting too is the symptoms aren't as severe as you have with Ebola virus or with Nipah virus, to where within 3 days, for example, when a person gets Ebola, they're debilitated and they're immobile. When you get COVID or you get anything, you get influenza, you're still able to function. You potentially can go to an epicenter and spread it some more. So if we look at cases where Ebola broke out or Nipah virus broke out, for example, in Kerala, India, where there were 22 cases, 21 out of 22 people did not make it. They succumbed to the virus, right? But we look at a place like India with a very high population, and we wonder, why didn't this take over like COVID? Well, the answer is simple. It just is that effective at killing people. So we look at a virus like COVID, which has a lower mortality rate, but it still makes you really sick. And then it kind of figured out, it figured out that it figured out the balance. It's able to inflict damage, but then make you just not sick enough to go out in town and think you're well, and then infect a bunch of people, come back home, get really sick, suffer complications, all while getting spread really effectively. So that's the thing that's really diabolical and really smart about respiratory viruses and what we need to tackle. The biochemical aspect of what makes respiratory viruses so smart is actually that they're faulty. They're not very good at replicating their own genetic material. So COVID is unique in that it is giant for a virus. It has lots and lots of genes. The respiratory viruses I study, for example, including, and then they're in the same order or the same kind of genetic, not family, but the same order. If you look at the phylogenetic tree, they're really closely related to Ebola. And these viruses have between 5 to 11 genes. That's it. And humans, for reference, have 200,000 genes. We have lots and lots of genes that encode for lots and lots of proteins. So we're really complex individuals. But then, you look at something really tiny that is submicroscopic with a fraction of our genetic makeup,
and it's kicking our butts.
Yeah, so we think we're pretty smart, but then we have this thing that we can't even see, and it's like, haha, I've got 7 genes, I'm gonna tackle you. So, so that's kind of the danger with viruses, is they're really, really smart, and they've optimized themselves to really carry very little loads, and in that they are really, really faulty in copying themselves. So if we look at coronavirus, for example, and this is going to be really surprising to most, but it's not as highly mutagenic as other respiratory viruses, because it has this phenomenon, and it has this ability to proofread itself, because it has, for a virus, again, it is giant. And so it has multitude of genes, and it's able to, when it's making copies of its genetic material, go back and kind of step back and be like, okay, so as Regina was mentioning, is this an A, is this a T, is this a G? Is this correct? However, it's not perfect, right? So even our systems, which are multifaceted in terms of, our systems as in human systems, which are multifaceted and are able to control themselves on multiple levels, we still are error prone. And so imagine a virus that has far fewer mechanisms of proofreading or of checking to make sure they copy themselves well. So this is what makes viruses, or respiratory viruses, particularly devastating, or particularly mutagenic and able to escape what we think they are. And so we look at COVID, for example, and recently, as many of us are aware, there's a Delta variant, which, if we compare that to the original Alpha variant, which was dominant maybe around this time last year, it is 60% more transmissible. It is just a beast. It's really everything we don't want about COVID. And the thing there is, because it's not great because it's so optimized for basically just carrying only what it needs and not anything else, because it's really small, even though it has proofreading activity, it's able to mutate, not on purpose. It doesn't do this on purpose. It just kind of happens, right? And then what happens in case of mutations is not all of them are going to be positive. Some of them are going to be deleterious for the virus. However, by natural selection, you're going to have the ones that survive and persist being the ones that are able to inflict worse effects. And so that's kind of what we're seeing as a result of not being able to get ahead of this virus and not being as careful, not masking up, not following up with vaccinations. And so this kind of highlights the importance of helping out everybody here by, not just us as individuals or just our families, but the entire population, the entire economy, if you're not thinking of it in terms of human lives, by just going out and getting vaccinated as much as we can ahead of possibly worse variants arising because of the biochemistry of these viruses just being really crappy at replicating themselves, resulting in possibly worse strains. what was the third part of the question? What I did for my dissertation. So what I studied was human metapneumovirus, which is the cousin of respiratory syncytial virus. So respiratory syncytial virus is, if you have any children or been around any children, you've probably heard of it. It's the leading cause of hospitalization in infants. And by the age of 5, most people, like 95% of the global population, would have had respiratory syncytial virus. And so it results in a common cold. But for infants who are preterm, like myself, I was preterm. When we get RSV, it tends to be a lot more severe, a lot more dangerous to the underdeveloped respiratory tracts of infants. Also, you get reinfected throughout life. And so for the elderly, it can also be an issue. And so outbreaks in nursing homes are also not very great. So HMPV, human metapneumovirus, which I worked with, is very similar. It was only recently resolved from RSV because the symptoms are very, very similar. And for those two viruses, there aren't great therapeutics. There's one for RSV, palivizumab, and then, so our lab worked on that a little bit. There's also another, and palivizumab, for the immunologists here, might probably recognize that it's a monoclonal or it's an antibody therapy, which means it is extremely expensive. So for people who have access, it's $10,000 per person per dose, which is cost prohibitive to say the least in our part of the world. So the goal there was to figure out the molecular details of RSV. So with RSV and HMPV, to figure out, well, what are the other targets? How can we develop a better vaccine? And kind of to piggyback off what Justice was talking about vaccine technology, in the 90s, there was a vaccine that was made, which was, I believe, based on the whole virus inactivated, formalin inactivated virus, and it actually was found to be worse, produce worse outcomes, which meant people had to regroup and think of, okay, what do we need to do? What did we do wrong here? What do we need to do? What do we need to develop in terms of a vaccine that would be potentially better and not result in worse outcomes? And the answer here was making palivizumab. And so as you were talking about developing an immune response, instead of giving the body a protein that the body would produce antibodies to, instead people just went ahead and made that antibody and then will infuse that into infants and then give them the antibody, give them those soldiers. So our work was trying to figure out, how do we make that antibody better in one sense? So that antibody was okay, but it wasn't optimized. So how do we optimize it to where it recognizes specific parts on the protein that it's targeting, to where it's able to more effectively, where you can use less of it to more effectively knock down any potential viral particle that's actually active. And then the other parts were looking at the biochemistry of the protein. So if you look at coronavirus, how it looks like it's studied based on the crown shape, those proteins are actually present in a lot of viruses that are enveloped, that contain a host-derived membrane from our plasma membrane. So a lot of those viruses have those proteins sticking out of them, and those proteins are what helps them dock onto our cells and what kind of opens our cells up. So a lot of our research was, how do we mess those proteins up to where they're not able to dock onto our cells and to undergo those biochemical changes that allow them to open up our cells and enter. So that was one part of my research. And it was a lot of biophysics and biochemistry, a lot of mutagenesis. So I did a lot of PCR, just like Regina, a lot of cloning with E. coli, a lot of, putting stuff in plasmids and protein purification and all of that. And all of that was to determine what regions of this crown or spike protein, or this, in our case, we just call it the fusion protein because that's exactly what it does. What regions of this fusion protein can we go in and do the least to, but mess up this virus on a large scale? So that was a part of the work I did. And then the second part was, okay, so let's say that this virus, for some reason, we're not able to target the outside proteins very well, and it's able to open up our cells and enter. What is it doing, and how can we? What are the effects of the virus in our host cell? So in our case with human metapneumovirus and RSV, what we found is the way it inflicts pathogenicity, the way it kind of makes us sick, partly relies in its remodeling of our cells. And so it takes our cells, which are, let's say, look a certain way like this. What it does then is it kind of stretches them out and reconfigures a lot of different signaling pathways, and then makes them fuse together. And so you're having your cells, it should be individual cells, kind of just joining together en masse, losing their functionality, losing, you know, their uniqueness and their specializations, and forming large multinucleated bodies that are called syncytia. So how do we prevent that? Secondly, another thing happening in infection was there were a lot of different things that we looked at, but another thing was the formation of these separated bodies called inclusion bodies, which were allowing for the virus to replicate without being bothered by the external cytoplasmic environment. And so it allowed it to evade immune responses by sequestering itself in a little corner in the cell. And then just, so another thing that we were really interested in was, how are these inclusion bodies happening? And this is kind of where in basic research, it may appear that we're kind of doing a deep dive just to do a deep dive. But the whole point of that is, okay, so now we know how these inclusion bodies happen. How do we mess them up? How do we go in and use, what kind of drugs are available right now? How do we go in and use these available drugs, or, you know, make a structure-based design to go in and interrupt the virus from being able to sequester itself in the corner and replicate effectively. So that was what my thesis was on. The earlier project I worked on was on the fusion aspect on those surface glycoproteins, or those surface proteins, and figuring out what makes them stable and what makes them able to be fusogenically or catalytically active. And the second part of my project was on what happens after a virus enters a cell successfully, and how do we circumvent it from continuing infection. I'm trying to remember the third part, if there was a third part of the question.
I think you covered most of it.
Okay.
Yeah, thank you so much. But it's interesting that earlier work on RSV has a huge bearing on the COVID vaccine as we have it. Because the reason that vaccine didn't work was the formation, the fusion changes. They realized that the RSV, of course, the virus changes its shape. So I think the team in the guy who was in Fauci's lab, who's now in Texas, found a way to add proline molecules to the structure. And that was what was used for the COVID vaccine that I think has made it so effective. That two extra proline molecules. And there are people who think that the fact that the AstraZeneca folks didn't add that mutation to their vaccine is a reason why it might not be as effective as the mRNA ones. So it kind of ties in to what. Thank you so much about that. And I think your quote is the quote of the day. I have 7 Gs and I'm going to tackle you. I love that. Let's go back to Aloysius. You and I talked a little bit yesterday about Burley ulcer and the spread of it. And one thing that the disease is known for is that nobody really knows how it spreads. I know there was an outbreak in Australia and they thought it was through mosquitoes and possums. But like you pointed out to me, if it was through mosquitoes, then everybody in Ghana should have it, right? Since we have mosquitoes everywhere. So in terms of the disease, what's the thought process about how it spreads? And what role does that particular toxin that Justice brought up, the mycolactone, play? And has there been any work into finding an antitoxin to mycolactone, which I think would be, to me, might be the best way, because it seems to be the central player in this disease.
Yeah, so thank you. So currently, what we know is that the mode of transmission, we don't really get a clear picture of the mode of transmission. Some aquatic bugs have been implicated in our parts of the world. The other thing is, those who live in the endemic communities have some peculiar, environmental factors. They live in places where there are water bodies, or places where the environment has been affected by, for instance, mining. That's why we have cases in places like Dunkwao. And then places where there are stagnant water like Tepa, and then some parts of Agogo. that is also one of the places where the disease is endemic. So what we know is that those who normally get it in those communities do work or farm in places where there's water bodies around, stagnant water and stuff like that. The other thing is it normally affects kids because they are those who are sent to go and fetch water in those villages. So that's one of the links that has been identified in relation to infection with the disease. In terms of family relation or infection related to families, we've had weird cases where people from a particular family will have a particular lesion around the same location, but nobody has really explored those scenarios. So maybe in the future, somebody will go into that. But apart from that, I can say that based on literature, the mode of transmission is still in confusion in the sense that we haven't really arrived at the correct mode of transmission. In terms of the liquid toxin mycolactone, it is cytotoxic and is also immunosuppressive. So the school of thought we currently have is that when people get infected with the disease, there's an incubation period, and then the organism, Mycobacterium ulcerans, produces the toxin. So some have shown that the higher the concentration of the toxin, the more immunosuppressed the individual is. Because Blue ulcer is technically an inflammation disease, so what you are expecting is high production of those inflammation markers. But what you rather get is, a suppression in production of those markers.
Do you see that more in people with HIV? Sorry, Aloysius. Would you see that more in patients with HIV?
So we published a paper, I think, in March on HIV co-infection. Actually, that was part of my master's work. And what we found out was that when you take Blue ulcer patients, you look at the immune responses of Blue ulcer patients and Blue ulcer patients co-infected with HIV. What you realize that there's when you do, let's say, basic ELISA assay, and you compare the immune responses between the two groups, those who have HIV co-infected with BU have a significantly lower immune response at baseline. So that is established knowledge. The other aspect of the disease is also paradoxical reaction. With that one too, what the expectation is that when somebody has a paradoxical reaction, it's also another form of hyperinflammation caused by mycobacterial antigens. So the expectation is that you are supposed to have a high level of inflammation markers produced like interferon gamma and the rest. But that one too, there's lower levels of immune response. That was from my master's thesis. So that's information too is there.
So that's all caused by the mycolactone, you think?
Yeah, so the suspicion is that it is caused by mycolactone. Because some of the researchers have also gone into basic sciences and have also shown that there is a blockage of the endoplasmic reticulum that affects translocation. And because of that, there's low production of Th1 markers, and some of those markers, that is what that's another school of thought that is existing in relation to that. So they are trying to say that mycolactone drives immune suppression and cytotoxicity in Blue ulcer. In my PhD work, what I'm looking at is the role of diet in the pathogenesis of Blue ulcer. And the question is, why am I looking at that? So in Rachel Simmons' work, they were able to identify that there is a depleted production of thrombomodulin, which is one of the cofactors related to the coagulation cascade. And if you go back, you realize that thrombomodulin production is related to vitamin K production. So the idea was that, is it possible that diet can have an effect on disordered coagulation or can affect the coagulation process? And my work is testing that hypothesis. So the first thing is to ascertain or to show that there is a problem in that situation, in the sense that do we have Blue patients who are malnourished, are we going to compare that to, let's say, match controls and see whether there's a dichotomy between the two groups? And so far, the work is ongoing, and there's a difference between the two groups. You can see that Bruliosa patients have malnutrition in a lot of micronutrients when you compare it to their matched controls. So the matched controls are people who live in the same communities, and some are even neighbors of the patients, and there's a slight difference. Some too, there's high difference in the nutritional adequacy levels. And protein, for instance, is less consumed when you look at the two groups. So there's an issue. We are dealing with diseases of the poor, and the assumption was that because they live in communities where they predominantly farm, the expectation is that they will eat food which are more healthy. But the situation is when you start to look deep into the picture, you realize that that is not what is really going on. So my work is really trying to break grounds with this aspect of Bruliosa so that maybe in the future we can look at other aspects of looking at metabolomics to understand how nutrition is affected, the metabolism of the patients is affected when you look at those aspects. So currently we are also looking at markers that have a link with those nutritional deficiency, because if you look at the synergistic role that exists between nutrition, immunity, and infection, is either malnutrition make you susceptible to an infection, or when you get an infection, does it, do you basically feed the organism to make you malnourished? So there's this synergistic role that exists between malnutrition and infection, and that is what I'm trying to play out for now.
I think it's been established that malnourished people tend to be more susceptible to infections. It just might be that they can't mount an immune reaction. It's interesting to see if you can see that with a BU. When they had an outbreak in Australia, they found out that the possum was actually sort of a vector for the disease, and that the droppings of the possum in the soil could be a way that people were getting the disease. Now, is that possible that there's a vector in Ghana that has not been found? We don't have possums in Ghana, I think, do we? I don't think, I'm not sure. But I'm wondering if there's a vector that has not been found. Do you think? I know you talked about the water bugs.
Yeah, so there's a published data that shows that people who get infected, some say they get this bite from a bug, and then after the bite, it develops into the nodule and then into the ulcer. So there's a high possibility that there is a bug or there is a vector in those areas that have not yet been discovered. And if you look at the terrain, you can see that we don't do a lot of research in those areas. I think if more work is done there, it will open more doors for us to understand what is really going on. Yeah.
What the Australians did, what was really elegant, is they did quantitative PCR on the possums, so they were able to, find those animals that had high levels of that. Now, has anybody done any work on an antitoxin to mycolactone that you know of?
Not that I know of. So what I know is, so for my MPhil work, what I did was we used the polyketal synthase domains that are responsible for the production of mycolactone to design subunit proteins, and then we tested it ex vivo to see if we will be able to, identify high responses. It was a case control study, so we wanted to see whether we can get candidates that can distinguish between the cases and controls and stuff like that. But we didn't really. We got high, better responses, all right. But I think more work needs to be done in that aspect. But for now, I haven't really, I've seen anybody done any work in relation to developing an antitoxin to that. But I think Justice can, if in case he saw something like that. Yeah.
All right. Thank you, Aloysius. So Justice, before I get to what I'm going to really ask you, any thoughts about anybody working on an antitoxin for BU?
Currently, I don't think there's anything like that in the literature, and I suppose there would be a reason why nobody's focusing on that. Even though we know the toxin is central to the pathophysiology, to the whole causation of the disease, it's also secreted by the bacteria. So if let's say someone catches an infection, the bacteria is in the skin or wherever, it's secreting the toxin. The toxin is causing the ulcer or the lesions you have. It is worsening it. Now, one interesting thing about the toxin is that it's very light sensitive. So with exposure to light, it breaks down, it disintegrates. So in fact, one of the proposed ways of treating Borrelia ulcer was to expose it to sunlight. And back in the day, a group, I think, developed some UV radiation of a sort, some UV light chamber where they were expected to put the infected area, and then the presence of the light was to cause integration of mycolactone. But the problem with that was that you still had the bacteria there. And once you had the bacteria, even if you disintegrated the mycolactone that was present, the bacteria was going to produce it again. So getting rid of the mycolactone alone was not going to be a solution.
It's not enough.
It's not enough. You still have to get rid of the bacteria. And that's why there was a lot of attention on antibiotics, and generating particular molecules or drugs which would tackle the bacteria and not necessarily the toxin. As when you clear the bacteria, you basically clear the toxin, even though the toxin stays on for a little while before. And in the lab where Jibo works, Aloysius works, our professor, my former boss, Professor Phillips, actually has data to show that the toxin actually outlives the bacteria, but it's over just a very short period of time, a very brief period of time, and it may not necessarily be the focus. But there are antibodies with neutralizing antibodies, or antibodies with the ability to neutralize the toxin. And I have actually tested a few in the lab as part of my work. There's a group that developed they sort of modified the molecule, and once you modify it, you're able to develop a toxin to it. The downside to it is that usually developing antibodies or testing for antibodies is easy if you are dealing with a protein. You just take your ELISA plate, you mobilize it.
It's a lipid.
Yes, but this is a lipid. So the conventional ways of detecting antibodies to it become very difficult. You have to do neutralization assays, which I use as a proxy. I developed an assay which I use as a proxy to measure that. It's almost impossible to directly to develop that. There's a group in Basel which attached a protein to it. They biotilated it, so they attached biotin, which is a protein sort of, so they could immobilize it onto ELISA plates for detection. But that also meant that they had to modify the structure of the toxin. So it wasn't mycolactone in the pure sense as you see it in a natural infection. And the antibodies they developed to that, or the antibodies they got through hybridoma, so they got the mice to secrete excess antibodies to these things. The antibodies were neutralizing. They're able to sort of clear the mycolactone and prevent it from working in cells when it was tried ex vivo. So you have your plates and you put the cells in there. Usually when you put mycolactone in cells, they will die. It kills them or stops them from working depending on the concentration of the mycolactone you put on the cells. But if you have the neutralizing antibodies in there, then because the antibodies are sort of I'm trying to find a very street word to use, very English word to use. I think neutralizing makes sense. The antibodies are neutralizing the mycolactone. The mycolactone is not able to exert its cytotoxic
effect,
or
effect on the body,
where it is either killing all its negative effects on the cells. So that was
now the disease is caused by mycobacterium. So of course we know mycobacteriums cause tuberculosis. So you would think that the BCG vaccine might have an effect, but it doesn't really help, does it?
It doesn't.
It doesn't.
Yeah, it doesn't. So I mean, it's still the baseline vaccine candidates for testing, but it doesn't. Work so far has shown that there's very little difference. It's a bit conflicting. In some work that was done, I think there's one in Uganda, some refugee camps, Rwanda and Uganda, and some refugee camps, they thought there was a difference, and BCG did something, which is the TB vaccine, did something. But later on, work has shown that it doesn't do much. And even in my mice work, I know that it does give some very slight protection, but it doesn't do much. It really doesn't do much. And the reason is simply because whatever be the case, you need to tackle the toxin. And the bacteria that causes TB, even though it belongs to the same group as the bacteria that causes Borrelia ulcer, does not
make a toxin.
Yes. So there's some variation in there. But even
so TB, let me get to the question I really wanted to ask you. So we've all seen how Cuba, a small, poor country, with not much, has got 3 vaccine candidates for COVID out. For people who don't know, Cuba has an amazing vaccine platform, and it's nothing elegant like mRNA vaccines or anything like that. They do subunit work just like you are doing. Is it possible for a country like Ghana to have such a program? And I'm not talking about having a factory that makes the vaccines that are made in the West. I'm talking about a program that develops vaccines based on maybe the construct you're working on. Is it possible?
It's very possible. I mean, it's very possible. The cost of making vaccines, I think the main cost or the bill comes in at the clinical trial stage. But with the laboratory work that develops the concept and that tries it in animals as well, it's not the most expensive thing. And it's very possible for Ghana to have done it. And as I mentioned earlier, there are very simple methods. My method is a very simple and straightforward method, not too expensive, very cost effective. And that's actually what Cuba is doing. They're using their home scientists, some they send outside, and they're basically doing this. They are having a problem scaling it up to production level, but that is another issue altogether. Once they have developed it and they have the patent, it's theirs, and they can go into all sorts of collaboration. But it was very possible, or it is still very possible, for Ghana to fund such work and to go into it. I know a few groups which have been involved in vaccine development for COVID, and it's not, the work is not too expensive. You just need the conceptual design. And especially after the Chinese people released the sequence for it, the Chinese released the sequence for the virus. It wasn't too difficult to pick out specific protein parts you needed. Like, we know the spike protein is crucial. Now everybody knows what the sequence for it is. You could very easily generate subunit proteins to it, put in your various adjuvants, just as has been done by Novavax, and which is actually basically what Novavax has done. And they've come out with a good platform. It wasn't going to be difficult at all. But, well, it's a matter of priorities, I guess.
Right.
And let me add that their success is not a sudden story. It's not because COVID came in and they suddenly went into it. It's something they've been building up over the period. So they built an industry for it, and they could capitalize on the skills and the technology they had at the time to come up with these things.
And they have nothing. I read somewhere where they have one mass spectrometer on that whole island. One mass spectrometer. I mean, and they are creating vaccines that are exported. And I think where the will, there is a way, and we could develop our own platform. Anyway, I want to open up the floor for questions, and I would like to go to Albert Kobina-Mensah. He wanted to share something about Buruli Alsa and the mining communities. Aloysius brought it up, and Albert wants to. Albert, wherever you are, unmute yourself. Let me find you, and I'll put the spotlight on you. Yeah, there you are. If you want to unmute yourself and make your submission. Albert, are you there? All right. Well, whilst we are waiting for Albert to show up, there are a few questions that have shown up, and Harry Akiglo asked this. He wanted to. His first question was actually to Justice, and it's based basically on what we just discussed, and it's about vaccine manufacturing in Africa. And he wants to know whether new technologies like Symbio has a role in enabling that, and whether we have capacities in Africa to do that.
I don't doubt we have capacity in Africa. I don't think human resources at all. In working in this field, I have met Africans, Nigerians, Ghanaians who have all sorts of skills. And the spectrum is not just people with pure vaccine backgrounds. You need microbiologists, you need immunologists. Sometimes you even need physicists, and how things work. So we do have the intellectual capacity. That much I don't doubt. We have the human resource. Unfortunately, you don't find them in Africa most of the time. They're out of the country or out of the continent. But if we did have the will to do it, we can easily gather the human resource. And India showed the way. If you've heard of the Serum Institute in India, which for me is a prototype of what Africa could easily have done. So the Serum Institute is basically a factory, a vaccine manufacturing factory. It was set up by the Indian government and some private sector. What they did was to say, look, in the West, they produce vaccines. These vaccines are quite expensive because over there, labor is expensive, electricity is expensive, and so on. But if we should set up a factory in Africa, in India, where because labor is cheap and all these things are available and are cheap, and India has a very big pharmaceutical industry, we have a huge population over there, which is already a market. We could get all these scientists and these pharmaceutical companies which have the intellectual property for the vaccine. So they develop it, they fund all the research and development. And once they have the IP, we go into collaboration with them to say, look, we have the factory set up and we have the scientists here. We brought our top-notch scientists from America, from everywhere in the world, all the Indians we know are great at this thing. We brought them back home and we've set up the factory which is able to supply, which is able to produce, and so give us the IP or give us the permission to be able to produce your vaccines for you and we'll supply the world. Before COVID, the Serum Institute was actually producing, I think, about 60% of vaccines worldwide. And they were doing so because they had a competitive advantage. So pricing, it's cheaper for them. They had the people at home and they were able to do that. Fast forward, when COVID broke out, as AstraZeneca and the group in Oxford were developing the vaccine, the AstraZeneca vaccine, the Serum Institute went into a sort of collaborative agreement with them and said, look, if you develop this and it succeeds, we can produce it for you. We know that there will be a lot of pressure on AstraZeneca to supply the world, but your capacity will not be able to meet it. So why don't you allow us to also produce the vaccine after you have developed it? And they'll definitely have their own terms and agreement. And we will supply to other parts of the world so that there won't be pressure on yours, and then you can focus on your UK population or your European population. And they agreed. So the Indians, through the Serum Institute, were actually producing the vaccines. And the COVAX initiative, or the AstraZeneca vaccines that were COVAX initiative, were from the Serum Institute. They were from India. They were secreted and they were producing these vaccines, selling them or distributing them to other parts of the world, and then keeping some for themselves. Somewhere along the line, they realized they needed a lot more. And so they tried to legislate against the export of the vaccine so that they could keep all at home. That didn't work. But when their figures got out of hand, it became obvious that they needed to keep the vaccine. So currently, Ghana, for instance, is struggling to get the AstraZeneca vaccines, even from the COVAX initiative, simply because India is not exported. And they have enough, they are producing enough to be able to take care of themselves before thinking about others. So countries have already taken ahead. They've been thinking about these things for years, and they've developed a platform, which is something Ghana can quite easily do if we had the will to do something like this. Research and development may be too expensive. It's a bit of a gamble, because the truth is that when people talk about drug trials or vaccine trials, the number of vaccines and drugs which enter trials and fail are actually more than those which get approval and get onto the market. So there's a very strict system to make sure that you don't get drugs or vaccines which are not good enough on the system or on the market. It's strict, it's stringent, except in a few places, which a country you mentioned initially, I don't want to go into that, where they may be a bit lax about things. But generally, it's not easy to get your drugs or vaccines on the market. It's a very stringent process, and most of them will fail trials. So you may not want to push your money into that. You may rather be at the production stage, initially gather some money before going into development. But for very basic work, like the immunology of it and so on, we can do it. And I know there's kindly work going on in Noguchi and KCCR in Kumasi on the vaccines and how they're working in the African population and so on. There was an African vaccine network. I fortunately do not know what came of it, but if we had the willpower, if we wanted to put our monies where our mouth was, Africa could very definitely come up with a candidate. It would not have been impossible for us at all. I don't doubt it.
Thank you. I think it has to be a continental thing.
A follow-up question to that,
sure.
Real quick, just if you will, or I guess an observation, which is the people are there, as you pointed out, Justice, and as Aloysius also was talking about. How do we convince funding agencies who have the money, the Bill and Melinda Gates Foundations, the pharmaceutical companies, I guess our own local funding agencies? Do we have a version of the NIH? Do we have anything like that? How do we convince them? How do we write grants? And how do we say, this is a tropical disease, it's a neglected disease, it's brutally ulcer. It is inexpensive to create subunit vaccines and pump them out. So how do we help ourselves here on our own continent, and also, I guess, exported help in terms of funding?
So it's unfortunate, but we don't really have any proper funding agencies in Africa or in Ghana. I mean, I've been looking for a few for a friend. I was looking through for a friend who's applying for PhDs, and we unfortunately don't have any strong funding agencies. All the money comes from outside, and the truth is that, with the exception of a few which are extremely philanthropic, most of these funding agencies have particular focus. Foci. So they're looking at, if it's cancer research, they're looking at, if it's neglected tropical, whatever it is they're looking at, they always have a focus. And they're not going to start up something new. For instance, there was COVID, funding agencies pumped out, the UK government pumped out money, the American government pumped money into the system through the NIH or through the UK, the NIHR, or through the Wellcome Trust or whatever. When they pump the monies out, they're looking for people who can deliver within a short period of time, and they're not looking for someone who is now coming to develop the technology from ground when someone already has a platform. So those who had developed the platform already had the advantage, and that is why African scientists could not have competed when it came to the COVID therapeutics and the trials and the funding. It was going to be impossible. You don't have the platform. Other countries have scientists which have developed these platforms, and all they needed was the money to get it going. They were going to get it. Going forward, we will need to start mobilizing resources to fund our scientists. We cannot always depend on the West. The West has interest. I was at a conference about 2 years ago, and I presented my work on my vaccine candidates. And at the end of it, I mentioned I had won a research grant at the time, and I mentioned that I've won a research grant and I'm transferring the technology into TB vaccines, and the initial results were good. At the end of the conference, one man from a funding agency, a very big funding agency, walked up to me and said, can we talk about your TB work? And I said, well, I want to talk about the Buruli side because that is almost complete and that is more of my focus. Yeah. And he said, well, how many people get Buruli ulcer disease in this world? Even if your vaccine is successful, what's the market value of it? And he asked a very realistic question. If someone is funding you to do this work, what's the market value? If they are funding work that is going to solve problems in Africa, and it's just an African problem, ultimately the Africans are actually going to come back to the West to come and help them pay for even the drugs again. So there's no commercial interest or value for them, and people are not pump money into such areas. They're going to keep pumping money into hypertension, into cardiac problems, into cancer, which are big Western problems. And that is actually why the WHO had to designate the name neglected tropical diseases, because they have really been neglected. Nobody's interested in these things, and it's a capitalist market. If you don't create the value for it, you're not going to get funded. And if you think it is important to you as a continent, then you will have to generate your own income and put it in there. Otherwise, you can't keep waiting for white people to come and do it. My former boss, Prof. Phillips, gets a lot of grants, and almost all his grants, all of them actually, are from foreign agencies, none from Africa. I think the AU has now started floating a few things here and there, but it's woefully inadequate to do anything very significant. And we don't have any fun.
I would like to add a little bit to that.
Yeah, sure, Lucius, go ahead.
Yeah, so as he said, that's the reality on the ground. I think based on the lessons from COVID, maybe it's high time as a continent and as a country, we start to look within. We have agencies, organizations, banks that are investing into other things. Maybe it's time they start looking at things like this, because nobody outside is going to, give you money to come and do a Buruli ulcer vaccine, unless, let's say, maybe climate change gets worse and then it's becoming endemic in other Western countries. But currently, because of the situation on the ground, it looks like we have to start looking within and solving our own problems. That's the little that I would like to add. Thank you.
Thank you so much.
If I may come in to add just a little, what many people do not, what many people, and I think many countries are not realizing, is that science is the new gold. You see, science is the new gold. It's money. Now, think about Pfizer. Think about a company like Pfizer and how much money they're going to be making from these COVID vaccines over the next 10, 15 years. Just monitor how their stocks have grown. Science is the new currency, is the new gold. Gold is going to get exhausted. You have exhaustible resources, your gold, your oil, and so on. But once you come up with something significant in science, you have the IP for it for many years. You're going to be eating of it, make so much. And there are people, generations, families which have not worked because some grandfather of theirs discovered something very significant, and that has kept the family for years. So it's the place to invest the money now. It's the place to start pushing now. It's going to improve on your industrial processes. It's going to improve your GDP. It's going to improve your human resource capital. It's going to improve your education. It's going to improve every single part of the economy. And if countries are not interested in investing, they will be missing out, unfortunately.
It's interesting you bring this up, but the essence of this, and I hope this turns into be a series. If you look at Ghana now, there's a very high unemployment problem among the youth. And it's because there are not very many industries that can employ these people. Unless you get a job with government, or you do galamsey, or you do agriculture, or you open a church, there's really not much to do. But in the whole field of science and technology, the opportunities are immense, absolutely immense. And it's high time our governments looked at that and diversified. The Asian tigers have taught us the way. And I'm hoping that I can continue with this series and talk to more young Ghanaian scientists and physicists and engineers and code writers out there to sort of put a spotlight on science, on STEM, basically, as the way into the future. Albert Kobina means, are you back? You had a little something to say about, so if you can unmute yourself and go ahead.
Thank you so much. Can you hear me, please?
I can hear you, yes.
Thank you so much. I joined the meeting courtesy of my very good friend, Justice. And I'm also a young scientist in Germany. And my research focus is into remediation of contaminated mine sites, especially from gold mining. I think that has been a very wonderful discussion, very educative, and I would like to make a contribution, one or two. Now, because I realized that there was a topic on Buruli Osa. And concerning Buruli Osa is an, I don't know if the people working on Buruli Osa had found out that environmental factors play a key role. And in Ghana, for instance, more than 1,200 cases of Buruli Osa have been recorded so far. Now, interestingly, all the cases that have been reported so far have been found in mining areas. So when you read the work with regards to Buruli Osa, many scientists are linking it to arsenic. It is because arsenic is found or is associated with the gold mine ore. Now, because this arsenic is associated with this gold mine ore, naturally, it undergoes oxidation or breakdown and it's released into the environment. And it gets into the groundwater, it gets into the soil, it gets into the ecosystem. At the end of the day, it is absorbed by the food chain. Now, the other way arsenic can also get into the environment is through anthropogenic factors, especially when gold mine is done indiscriminately or unsustainably, and it leads to exacerbation of the problem. Now, arsenic has been found to provide a surface or a fertile ground for the growth of the bacteria that causes Buruli Osa. And it is also because Buruli Osa is one of the cancerous skin diseases. And arsenic is noted for skin disease. And when you go to Bangladesh, they have a major problem with arsenic pollution, especially with regards to their groundwater. And cases of Buruli Osa and other sources of kinds of skin diseases are all in abundance. Now, I heard one of the young scientists speak about the disease can be spread through genetics. Correct me if I'm wrong, but that's what I heard. Now,
there is a genetic predisposition to that. Yes.
Thank you so much.
Maybe, yes.
Yes, and my brethren, it is very true, and it is established by science that many of the environmental diseases like Buruli Osa and even Parkinson's disease and other other cancerous diseases and other nervous diseases that affect the nervous system. These diseases are capable of being transferred from the parent to the offspring. Now, a typical example of this disease is it's Parkinson, stroke. All these diseases are genetic. Now, let me end here. Link this disease to, let's say, copper. You know, copper is released into the environment from mining and from other anthropogenic or natural sources. Now, copper is responsible for Parkinson disease. It is because this copper, there's a gene, I think it's called ATP7B or so. This gene is responsible for regulating copper in the body. Now, when this gene is denatured, it is capable of transferring this disease from the parent to the offspring. And the same thing also happens with the buruli or with any other environmental diseases that can accumulate from in the mother, and it can be transferred into the onto offspring. And so many sources through our drinking water, our food that we eat. I end by saying that as a country, we need to take keen interest in managing all resources using science methodology that works. Science methodology that works. When you go to gold mining areas in Ghana, people are suffering from all sorts of diseases. And my brother, they don't even know that the sources of the diseases are coming from the environment where they are living.
Yeah, that's so true. That's so true. I think what you're trying to hint at, and I'll let Aloysius touch on this, and also let Regina touch on that. It's the role of heavy metals. Arsenic is a heavy metal. Copper is a heavy metal. Is the role of heavy metals in disease, and that in some of them, they can cause diseases that mimic known diseases. For instance, mercury has an effect in the brain and causes a disease that looks a bit like autism. The heavy metals can also lead to mutations, like you mentioned. So if arsenic plays a role in the development of Buruli ulcer, it's possible. And I'll let Aloysius touch on that. Thank you so much. Aloysius, what do you?
Thank you so much. It's a pleasure.
You're welcome.
So as I said, we have one clinical site at Dunkwao where we manage patients. We do get people who are galamsey operators and people who are basically into mining. We are aware that there is this paradigm or school of thought in relation to arsenic. So it is possible that could be the main cause for the spread in those areas. But there are also other areas where they are not doing mining activities, where the disease is endemic. So we need to look at both sides of the coin and understand, appreciate how things work on those other sides. So for the arsenic story or the arsenic paradigm, we know and we are aware of that. And it's also possible that could be the reason why. But when you go to other places like Agogo, communities that are way, way, way far away, they are not really doing any mining activities there. It's just people who are doing small scale farming. So what do you say for that too? So that is what I'm aware of. Thank you.
Yeah, all right. So Regina, let's come back to you and talk a bit about the role of heavy metals, and then there's a question about the ethics of CRISPR that was asked a while back, and I would like you to touch on that too. We have about 15 more minutes on the program, so let's try and squeeze in as much as possible.
Okay, so I'll just make a small comment about what the gentleman said about the mutations. Actually, mutations can be either somatic or germ cell, right? Somatic mutations, these mutations occur in all cells of the body, except the reproductive cells, right? However, the germline mutations, they occur in the sex cells, the gametes, So these are the mutations which can potentially be passed on to progeny, to offspring. It's not just, I just wanted to make that correction in the man's submission. Yeah. So back to the CRISPR. Yes, you wanted to know about the ethics, right?
Ethics of that. There's always that worry.
I think, when it comes to CRISPR, and I'm reading some of the comments on Facebook, and there is someone who is particularly concerned about the application. Well, I think we should not lean into fear, okay? I mean, science, I always say science will keep sciencing. You can't stop science. It will keep advancing, technologies will pop up here and there. What we can do is to regulate it, and that is what the researchers, the inventors are actually doing. I think 2 years ago, Jennifer Doudna, the American Nobel laureate, she came to my school to give a seminar on this CRISPR-Cas9 technology. And she talked about trying to actually have an international body, to look at the rules and regulations and the ethics surrounding, the use or application of this technology. So yes, CRISPR is a very powerful tool, and I think people are quite concerned. I saw someone talking about, where do we stop? Are scientists trying to play God? And with this potential, we can, in our field, we talk of the potential to create designer babies, which is quite a possibility and a reality with this technology. You can basically edit out what you don't like in the fetus, or introduce what the traits you want a fetus to have. These are all possibilities. But as I said, the scientists are working really hard to make sure that there is an international rule or body that regulates this application. So yes, but my concern is that we shouldn't really lean into fear. We tend to fear technology. I don't know why. Even with mRNA, it's just fear. We just lean so much into fear. So let's not fear the technology, but however, let's.
Do you remember the Chinese researcher who
yes,
fiddled with babies a few years ago
and made them
up
HIV resistant? Yes, and I think
yes,
I
exactly.
saw that, and it's not just me. Everyone in the scientific community was appalled by what he did. He was just trying to show up, like say I'm the first person to have done this, and because it was quite unnecessary what he did, right? So yes, yeah. I think the scientific community, we are all in agreement that this technology needs to be monitored, regulated, and that is what scientists are working on at the moment. So yes, hopefully in the future we'll have stronger ethics and guidelines on the use of the CRISPR-Cas9 technology.
Okay, all right. So Jean, any thoughts to that about the ethics of CRISPR?
Absolutely. I think what when we think of the unethical uses of CRISPR, we think of the person who edited the human genome, right? But we also have to focus on the good that is bringing. A lot of times when we think of scientists, we think of, unfortunately, sometimes the portrayal of somebody who is diabolical, who's going to do something for evil. But if we look at science on a large scale, from the invention or from the discovery of penicillin to chemotherapies and different therapeutic modalities, we need to focus on how those are benefiting our community. We also need to focus on the fact that there are currently a lot of laws. For example, you can't have gain of function viruses, mutations and viruses. You can't, there are a lot of things governing people, and a lot of our motivation to do science is not to damage people. A lot of scientists out here are actually really working for the greater good, and so it's a two-way street where we need to develop the trust of the community by being transparent about our science and by explaining it in the best ways possible, avoiding jargon. But it also only works if people believe us when we say, no, we actually don't have an interest. It's actually really difficult to get science to work in the first place. And so just trying to use it for evil is not only policed heavily, but also people just don't want to do it.
Yes, and I would like to add a bit to what Jean said, okay? So to understand diseases, first of all, we need to be able to study them in the lab, right? And that is how powerful this CRISPR-Cas9 technology, it allows us to create these models to study diseases. We can't just come out with drugs that work miraculously. We need a model to test these things on. For instance, in my case, if I develop something interesting in myopia, I'm not going to test it on Nana Dazi, who has myopia. I should be able to test it in the lab, right? And on the technology of gene editing, before CRISPR, we had tools to edit the genome. We had the zinc finger nucleases, we have talents. But the problem with these technologies was the specificity of the editing, right? You edit, and then there is what we call off target or off-site, editing occurring as well. But CRISPR is so specific. The specificity is so good that it edits exactly what you want. And with this, we can create much powerful, models, disease models to study in the lab. So yes, as Jean rightly said, we shouldn't really look into the negativity, and try to, be optimistic about the whole the positive side of the technology, and not lean so much into the negativity associated with it. Yes, that's all.
Yeah, thank you so much. There's a question here that says, should the first response to pandemics always be therapies to the neglect of diagnostics? Should the two not be happening at par? And I think we have enough diagnostics, right? I mean, Justice, wouldn't you say that?
Yeah, so it's quite interesting. The person asking this must be a scientist or someone with a lab background. But the dynamics are such that you first need your diagnostics. So without your diagnostics, it becomes very difficult to even go into therapeutics, because who are you going to treat? So it's usually hand in hand. The good thing with it is that, as it turns out, a lot of the diagnostic work actually feeds into treatment. So it feeds into treatment. A very good example is for instance, the development of identification antibodies during COVID. So during COVID, initially the idea was to find antibodies which we could use to diagnose, COVID, which are specific. If you find antibodies, are the antibodies specifically due to COVID, or could they be due to other viruses as well? And in searching for this, if you find accurately diagnose COVID, is there feedback? I'm getting feedback. Okay, it's okay.
Oh, that was me. Sorry, I apologize.
Okay. So in searching for the antibodies which would be very specific for COVID, we also found that the spike protein, which is a part of the virus, produced a certain antibody which was not just specific for COVID, but was also very good in terms of conferring protection or neutralizing the virus. And that is how the talk of the vaccine actually came up. So it goes hand in hand. In looking out for diagnostics, you also have to focus on treatment, and it's impossible to make them mutually exclusive. They'll always have to go hand in hand. The balance sometimes becomes a problem, because people are interested in, let's cure it. They're not really interested in how to properly identify it. There are still conditions which we have cures to, but the diagnostics are still a bit shaky, because it could be so many other things. It could be differential, but we still go ahead to treat anyway, and people are fine with that. Once there's treatment for it, they are happier. But going forward, I think we do know that blind treatment is also a problem, and you'll always have to get your diagnostics right. It will not be proper to put one ahead of the other. They should always go hand in hand.
All right. So thank you all so much. I think we have 5 minutes. The Hearts Kotoko match starts in 5 minutes, and I want to get you all off so you can go watch it. And so I'll just open the floor for closing remarks. Anything you want to say to the youth in Ghana about getting into the sciences, what you see as a future in your field or in the sciences? And let's start with Jean.
Yeah, so I mentioned earlier, growing up, I just didn't have a lot of examples of scientists who looked like me. So it was a little bit like, I felt like I was blazing the trail a little bit as a scientist, as a woman of color, especially for my degree. I'm the first woman of color to get my degree from my university, to get a PhD from my university. And so it definitely looked, and somebody, there are many times where you can walk in a space or you can speak a certain way and people would assume your intelligence based on the way you look or some kind of implicit bias they may have. Please do not let that discourage you. Follow your science and follow it fearlessly. I'm a strong proponent in that. And the four of us, this is what scientists looks like. This is the future of science. This is the present of science. So if you're watching this and you think a scientist should look or sound or whatever a certain way, it doesn't really matter as long as you have a passion for it. Please never let that stop you. And definitely reach out if you have any questions. But even small girls like me can do science successfully. So
Thank you so much. I like that. this is how science looks like. I love that. You've given me 2 quotes for the day. I have 7 kids and I'm going to tackle you. That's really my favorite. Yes, sir, Regina. What are your closing remarks?
Yes. So to anyone watching, as Jim rightly said, just go for it if you're interested in the sciences. Yeah, the chemistries, the physics. I always advocate for people to study the pure sciences, not just medicine, pharmacy, or engineering. Go for the chemistry.
Hey.
I know. The biochemistry, the biology. And to me, when I started, I remember when I got admitted to KNUST to study biochemistry, someone just told me, what are you going to do with a degree with biochemistry? You're either going to end up as a teacher or unemployed. And thank God I didn't listen to them. I mean, you know how when you get admission to the university, you're happy, you're excited. I was literally in tears because like, what am I going to do with a degree in biochemistry? But here I am now. So please, don't let your present circumstance just limit you or define you. I mean, I never thought I would be here. I love studying. So all I did was to study, and here I am now. So go for it. And I would just like to say, I hope our leaders, they have a change of mind and invest in the sciences, in the biomedical research. In my field, for instance, I get scared when I think about the prospects of biomedical research in Ghana, because we face what we call an explosion of technologies. The techniques I'm using right now, the next 5 years, they'll be obsolete, right? And I ask myself, when will Ghana even get these technologies? When will they reach Ghana? When will Ghanaian scientists even use these technologies? So I pray that our leaders invest in the sciences. Not just our leaders, our universities can also offer scholarships to students, our smart minds, our bright students. We can offer them scholarships to study research, PhDs, not just, we don't all have to move outside to do PhD. We can do it in Ghana as well. So these are the things I hope to see about the future of biomedical research in Ghana. Yes, so
thank you so much. So Aloysius, your closing remarks?
Thank you. I would like to say that there is this, a huge opportunity to solve problems in Ghana. And I would like to entreat anyone who wants to go into biosciences to really go into it. There are opportunities at KCCR in Kumasi. If you are in Accra, there are opportunities at Nguchi. And I think it's a good thing to do research which has an impact. I mean, Justice and I were part of a research where, after the clinical trial, Reliosa treatment was, changed to an all-oral treatment, which changed policy. It was a WHO drug trial. And things like this are happening through little efforts that we are doing here. So I would like to advise everybody or anyone who wants to go into sciences that it's a good place to come to. You are likely to make an impact. And I'm also hoping that our leaders, as Regina said, will start to look at their priorities and start to prioritize on science. Because if you're a country and you focus on science, there is a possibility that your nation would transform rapidly. We already have evidences of that in the Asia and then other parts of the world. We don't need God to come and tell us that this is what we need to do. It's obvious. So I'm just hoping that our leaders will, prioritize and then get things, changing. That's all.
Thank you so much.
Thank you.
So, Justice, your closing comments? Unmute.
Sorry. I was stealing church time, so the sounds from church comes up because I'm on top of the church building. Sorry, that's like yeah. So they're encouraging people to get into science, which I think is brilliant and which I'm always very happy and enthusiastic about. I originally wanted to go into the sciences straight. My interest was in the mathematical and physical sciences. But at the time when I completed secondary school, it wasn't sensible to tell your parents you were going to go into pure science or physics or math. if you had certain grades, you had to go straight into medicine or some top engineering. You know. So I ended up in medicine. Finding myself in medicine, I still thought, look, this is good. This medicine gives you some comfort and security, but medicine is based on the science, and the science itself. You need to understand the science to be able to be a good doctor and to be able to, you know, produce good medicine. And so I decided to go into the sciences on the encouragement of others. But a key thing I want to talk about is mentorship. You know, get a mentor. You always need to have someone you look up to and someone who will guide you. The step you are taking is not new. You're not the first to take that step. Someone has been there before. Someone has made the mistakes you are going to be making. And it's important you have someone telling you, don't go here because when I went there, I made this mistake. Don't do this. Get a mentor. Don't sit in isolation and think you can do it. It's always important to get yourself a very, very good mentor who will lead you through. Also, times are changing. The days when, as Regina said, people thought everybody who went into science ended up in a low-paid job, those days are over. Argue it out, make your point clear. If you want to go into the sciences, science is the future. It is the currency. There are lots of PhD opportunities outside the country. There are lots of master's opportunities outside the country. If you're interested, reach out to people. Don't be shy. Don't be withdrawn. Don't be subdued. Send emails around. You know, someone will respond. Some PI somewhere will respond. You may get into some life transforming opportunity. And as much as I hate to do this, I think I'm forced to do it. Life is not only in Ghana. Be open to the idea of having to leave your comfort zone if you have to, because the truth is there's a lot happening. The pace is fast out there. And if you really do want to get on top of the sciences, then you may unfortunately have to leave. And if the environment is not providing what you need, the support you need to go get it elsewhere. You can come back. The opportunity will always be there for you, but don't be afraid. Don't feel subdued. Get a good mentor. Move into the sciences if you are interested. You will be comfortable. And scientists are not, you know, they are not the people with the beards anymore. They are not the people. They have flashy lifestyles now. They live pretty good. They live a very good lifestyle. They party. They do everything like everyone else. You don't have to be a nerd to do it. You don't even have to be super smart to do it. You just need to be someone with interest and with a curious mind, and you will be successful at it. As leadership and pushing money into and investing into sciences, I think, you know, we don't need to say anymore. They know what they need to do if they are interested in doing it. Thank you.
Thank you all so much for doing this. I really appreciate that. When I reached out to each of you, you so willingly were ready to do this. And the others that I reached out to that, you know, didn't work out one way or the other. Thank you to you too. I'm sorry I couldn't ask all the questions that our audience wanted asked. That the time was our enemy, but I think we touched on a lot of things, and I hope to continue with this series, and maybe we can do it again another time and talk about other things. Thank you so much. And Regina, especially to you, I know it's like, what, 11 p.m. over in Hong Kong, and you have to be up tomorrow to go to the lab. Yeah, thank you so much for doing this. Thank you, Jean. I know you just moved and you're trying to settle in, and Justice and Aloysius too. Thanks for everybody who listened in, either with us here on Zoom or on social media. Thanks for your time on a Saturday, and go Hearts, phobia. So bye and have a good day, everybody.